Welcome to The Explainer. Today, we are going to decode the truly incredible mechanics of biological nitrogen fixation. We're going to break down this entire process, starting all the way up at the macroorganism level and then zooming right down into the microgenetics that actually make it all possible. But before we get into the nuts and bolts of it, let's talk about why this actually matters to you. Because quite literally, without the process we're looking at today, there'd be no amino acids, there'd be no proteins, no DNA, and ultimately, there'd be no us. It is the absolute fundamental biological bottleneck of all life on Earth.
Think about this for a second. We live on a planet completely surrounded by an atmosphere full of nitrogen gas. It makes up nearly 80% of the air we breathe, but it's essentially inert to most life forms. Those nitrogen atoms are locked together by an incredibly tough triple bond. We're talking about one of the strongest chemical bonds in nature. I mean, even the intense heat and energy of a lightning strike struggles to break it apart. Yet, somehow, microscopic life does it quietly every single day. So how exactly does nature take this unyielding gas and transform it into the usable ammonia that sustains plant growth and, by extension, every single living thing on Earth?
Okay, let's establish our baseline here. Biological nitrogen fixation is defined simply as the conversion of atmospheric nitrogen gas into ammonia. But what is honestly so fascinating about this is that this process is entirely exclusive to prokaryotic organisms. Now, a prokaryote is just a simple, single-celled organism that lacks a distinct nucleus, your basic bacteria. No advanced plant or complex animal can fix nitrogen on their own. It's a massive biological irony when you think about it. The most advanced towering redwood trees and complex mammalian predators rely entirely on the absolute simplest, oldest life forms on the planet just to survive. We aptly call these essential microbes nitrogen fixers. Here is our quick roadmap for today's explainer. First, the nitrogen fixers. Second, the nitrogenase enzyme complex. Third, the oxygen problem. Fourth, the molecular dialogue. Fifth, infection and nodule formation. And finally, genetic regulation, NIF and fix. Let's dive in.
Section 1, the nitrogen fixers. We'll be looking at free living and symbiotic partnerships.
So the incredible microbes pulling this feat off basically operate in one of two ways. On one hand, you've got independent free living fixers. Think of these as independent biological fertilizer factories. They just float around in the soil or water, doing the heavy lifting all on their own. This group includes cyanobacteria like nostoc and aerobic bacteria like azotobacter. On the other hand, we have symbiotic relationships. Now, these operate a bit more like exclusive corporate partnerships, where the bacteria team up with a very specific host. We see this in the classic pairing of legumes with rhizobium bacteria, or even the tiny aquatic fern azolla partnering with anabina. It just goes to show that whether they're operating totally solo or locking into intimate partnerships, biology always finds a way to tap into that atmospheric nitrogen. Moving on to Section 2, the nitrogenase enzyme complex. This is the molecular engine of the whole operation. If we zoom way down into the molecular level, we find this elegant two-part structure making up nitrogenase. First, you've got the smaller Fe protein, which contains an iron-sulfur cluster. Ferredoxin donates electrons to this Fe protein, which then hydrolyzes ATP to power up the second component, the much larger MoFe protein. Now, this massive 180 to 235 kilodalton protein with its complex molybdenum iron-sulfur clusters is the actual assembly line. It takes those electrons and physically forces the nitrogen gas to reduce into ammonia and hydrogen gas. But here's the catch. This molecular engine comes at a huge cost. Breaking that stubborn triple bond requires about 16 molecules of ATP to reduce just a single molecule of nitrogen gas. It is an insanely expensive biological operation, requiring just a massive amount of cellular energy.
Which brings us to Section 3, the oxygen problem. This is the great biological threat to the system.
Okay, we run into a massive, kind of mind-blowing biological paradox right here. Oxygen is the very element that powers almost all complex life on Earth, right? Yet it acts as a deadly poison to the very engine feeding the planet. In the presence of oxygen, that smaller Fe protein we were just talking about is irreversibly inactivated in merely 30 to 45 seconds. It's practically a ticking time bomb. Even the larger MFE protein has a half-life of only about 10 minutes in regular air. If a single breath of oxygen gets into this mechanism, the molecular engine basically rusts out and shuts down for good. So, how do they survive this? Well, nature engineered some genuinely brilliant microanaerobic solutions.
First off, free-living cyanobacteria like Anabaena create specialized cells called heterocysts. You can think of these as completely oxygen-free panic rooms that lack the oxygen-generating machinery of normal photosynthesis. Next, bacteria like Azotobacter take a completely different route. They just crank their respiration rates up to extreme levels, essentially burning up the oxygen before it can do any harm. Others use temporal separation, basically choosing to only fix nitrogen in the dark of night. And finally, in symbiotic plants, we see the development of nodules filled with leghemoglobins. These oxygen-binding proteins are evolutionary cousins to the hemoglobin in human blood. That's actually why the plant nodules literally bleed pink when you cut them open. They act like a perfect biological buffer, keeping free oxygen perfectly dialed in at a super low 20 to 40 nanomolar concentration. Safe for the nitrogenase time bomb, but just enough for the bacteria to still breathe.
Section 4. The molecular dialogue. How exactly do they establish this symbiosis?
When nitrogen is scarce, host plants and bacteria have to negotiate this complex partnership without triggering the plant's aggressive immune system. It acts like a highly secure, diplomatic chemical handshake. First, the plant root sends chemical attractants called flavonoids out into the soil. The rhizobia bacteria detect these signals, which immediately turns on their regulatory NOD gene. This, in turn, sparks the common NOD genes, NOD A, NOD B, and NOD C. Working together, they synthesize incredibly specific signaling molecules called NOD factors. Now, chemically speaking, these are lipokitten oligosaccharides, but you can really just think of them as an incredibly complex, multi-layered cryptographic key. The structure is so precise that only the exact right biological password will allow the bacteria entry into the plant root.
Section 5. Infection and nodule formation. Literally building the microhome.
Once the plant verifies that cryptographic password using specialized root receptors, it opens the gates for what's essentially a highly welcomed invasion. First, the plant's root hair physically curls right around the bacteria, deliberately trapping the rhizobia. Then, an infection thread forms, driven by the fusion of Golgi-derived vesicles. This creates a microscopic, custom-built highway heading straight down into the root. Meanwhile, deeper inside the plant, cortical cells start dividing rapidly to build the foundation of the nodule. Finally, the infection thread reaches this new structure, and the bacteria are released. But they aren't just dumped into the cell. Oh no, they are safely wrapped in a host-derived membrane called a symbiosome. Inside this custom VIP suite, completely pampered and protected, they differentiate into their final nitrogen-fixing form, bacterioids.
Finally, Section 6. Genetic regulation, NIF and Fix. These are the master control switches.
To truly master this biological ballet, we have to look at the master switches down at the DNA level. It's crucial to distinguish between the two main groups of genes running the whole show. The NIF genes are the core code required for nitrogen fixation. Think of them as the construction crew. They physically build the nitrogenase enzyme complex, and you'll find them in all fixers, whether they are free-living or symbiotic. On the flip side, the fix genes are specific to the symbiotic relationship. You won't find them in free-living bacteria at all. If the NIF genes are the construction crew, the fix genes are the advanced HVAC system. Their entire job is to monitor and strictly manage that unique, potentially deadly low-oxygen environment inside the plant nodule.
So how does the HVAC system talk to the construction crew? Well, this genetic regulation acts exactly like a line of biological dominoes. It's an elegant if-then sequence. If oxygen is low, then the fix cell hemoprotein senses it and activates. If fix cell activates, it acts as a kinase to turn on FixJ. If FixJ is activated, then it switches on transcriptional regulators like NIF-A and FixK. Ultimately, that final domino signals the NIF genes to start building the nitrogenase engine. It's brilliantly simple. The system absolutely guarantees that the cell only spends its precious energy building the highly sensitive nitrogenase enzyme when the environment is 100% confirmed to be safe from toxic oxygen. I want to wrap up this explainer by having you think about the future for a moment. If we can truly master this intricate genetic ballet, the diplomatic chemical passwords, the oxygen buffering, the precise computer-like code of the NIF and Fix gene cascades, we might one day engineer non-legume crops, like wheat, rice, or corn, to fix their own nitrogen. If we actually pull that off, we could eliminate millions of tons of synthetic nitrogen fertilizers. We could halt the devastating agricultural runoff that's destroying our oceans. We could fundamentally rewrite global food security for billions of people. It's a totally world-changing prospect, and it all stems from deeply understanding these incredible, invisible, microscopic bugs. Thanks so much for learning with us today.